Welcome to another installment of Gut Health in the News. Here's what I'll be covering today. Researchers from Monash University in Australia have discovered a link between high-fat diets and the onset of obesity and insulin resistance, and a possible therapeutic target to help reverse these conditions. Research out of Michigan State University has identified a link between specialized cells in the gut and inflammatory bowel disease, which could lead to more effective treatments. Researchers from the University of Washington School of Medicine have published a study linking certain types of bacteria in the gut to the formation of polyps and colon cancer, providing another option for screening and early detection. And finally, scientists from the University of Texas Southwestern Medical Center have discovered how vitamin A enters the immune cells in the intestines, with a potential for improving digestive diseases and vaccine efficacy. We'll have more on these stories right after this. Monash University researchers have shown for the first time that mesenteric or gut lymphatic dysfunction is a potential cause of and therapeutic target for obesity and insulin resistance. As shown through preclinical models, a high-fat diet stimulated the disorganized formulation of new lymphatic vessels in the gut. These vessels tended to leak lymphatic fluid into the internal or visceral fat tissue in the abdomen, triggering the promotion of insulin resistance. Quoting the article, "In this study, we were able to uncover a biological reason behind why the accumulation of fat around the abdomen is correlated with higher rates of metabolic disease, such as type 2 diabetes, than accumulation of fat in other regions of the body. Using a groundbreaking technology, researchers were able to deliver a COX-2 inhibitor directly to the gut lymphatics, which reversed the changes, restoring normal function and substantially reducing weight and insulin resistance." Work continues to develop new therapeutics based on these findings. For more than 3 million adults in the United States that suffer from inflammatory bowel disease, or IBD, the very food we need to survive is also a daily source of pain and discomfort. While no cure exists for IBD, new research led by Michigan State University is bringing fresh insight to the table. They've discovered an unexpected connection between specialized cells in the gut called glia and the defective genes involved in IBD. Hundreds of millions of neurons line the digestive tract to make the enteric nervous system, and glial cells surround each one. Not only can glia communicate with other cells, these cells are critical to proper immune function to protect gut neurons from damage due to inflammation. Genetically defective genes lack the ability to fully express an antigen known as major histocompatibility complex class II, or MHC2 for short. This results in a suppressed ability to control inflammation around the neurons in the gut, resulting in pain and deterioration of the intestinal lining. With this new understanding, it is hoped that more targeted treatments can be developed in the future to help people suffering from IBD. In a study posted in the journal Cell Host and Microbe, scientists from the University of Washington School of Medicine report the increased presence of a type of bacteria called Bacteroides fragilis, or B. fragilis, in the biopsies of patients with colon polyps. Upon closer inspection, the researchers found that these bacteria differed in their ability to induce inflammation when compared to the same strain from polyp-free individuals. Quoting the article, "What our data suggests is that in order to survive with an environment where metabolic and inflammatory changes are occurring, a normally healthy gut and related bacteria may adapt in such a way that causes it to contribute to the inflammation rather than suppress it." Colorectal cancer is the third leading cause of cancer in the United States, and its incidence is rising among younger adults. If a screening were available to test the microbes before a polyp even appears, it could be a key factor in driving these rates down. Vitamin A, a fat-soluble nutrient that the body converts to a molecule called retinol and then to retinoic acid, is important for every tissue in the body. It's particularly crucial for the adaptive immune system, a subset of the broader immune system that reacts to specific pathogens based on immunological memory, the type formed by exposure to disease or vaccines. Immunologists and geneticists at University of Texas Southwestern Medical Center have discovered how vitamin A enters immune cells called myeloid cells in the intestines. These findings could offer insight to treat digestive diseases and help improve the efficacy of some vaccines. A protein called LDL receptor-related protein 1, or LRP1, was discovered to be the mechanism for transport of retinoic acid into myeloid cells, and deletion of the gene responsible for the expression of LRP1 resulted in a virtual elimination of the adaptive immune response to intestinal infections. For inflammatory diseases such as IBD and Crohn's disease, tamping down the immune response via this mechanism could provide effective relief. Conversely, enhancing this process could boost immune activity, making oral vaccines more effective. That's all for this episode of Gut Health in the News. Please like and subscribe to continue receiving great content that hits you right in the gut.